Hello, and welcome to BioTech Talks, a podcast that dives into science, stories, and conversations around biotech and genetic engineering. Produced by BioTech Insights in collaboration with Oregon State University's College of Forestry and funded by the National Science Foundation, this show is here to make biotech feel less intimidating, a little more human, and way more interesting. I'm Tegan McGilligan. I study design and innovation management here at Oregon State. I'm a photographer, a videographer, and an art director, so I figured, why not bring my creative background into the world of podcasting? This show is a chance for me to use what I know, visual storytelling and design, to explore something I don't know much about, biotechnology. I'm here to learn alongside listeners and ask questions people might be too afraid to ask. And here's my co-host, Mary Long. Tell us a little about yourself.
Hi, I'm Mary Long. I'm studying digital communication arts, marketing, and sociology here at Oregon State University, and I'm really excited to join Tegan here as a co-host. I also run the TikTok and Instagram accounts for the BioTech Insights brand. Each week, we'll talk to scientists, students, researchers, and guests from all kinds of backgrounds, breaking down everything from GMOs and CRISPR to forest genetics and food systems, and the ethical questions that come with all of that. We're also building out short-form content on TikTok and Instagram, so if you're more of a scroller than a listener, we've got you there too.
So whether you're deep into science or just biotech curious, this is a space to explore, question, and get a little closer to the tech shaping our future. We're glad you're here. Let's get into it. All right, welcome back to BioTech Talk. Today I am joined by the In the Woods podcast, hosted by Scott Levengood, as well as Steve Strauss. Scott, would you like to introduce yourself?
Sure. Yeah, thanks, Tegan. So as he said, I'm Scott Levengood. I'm with the Forestry and Natural Resources Extension Program at OSU. And one of the things we do is this In the Woods podcast. And we have the pleasure this episode of having a chance to talk with Dr. Steve Strauss, who is, I'll get his title right here. He's one of my colleagues in the College of Forestry, and he's a University Distinguished Professor of Forest Biotechnology. So what do you say we start with an introduction? Always a good place to start. Steve, tell us a bit about your background and the primary areas of focus of your research.
Yeah, thank you, Scott. Thanks for having me here. Much appreciated. So I grew up in Brooklyn, New York, so I'm a city boy. And over time, I got really interested in nature and trees and started doing some backpacking. And that led me to ecology, and that led me to forestry. So that's how I got here. And then over time, I got interested in the field of genetics because the science was exploding with possibility and was interesting. And then people started growing hybrid poplars in Oregon, and they happened to be really amenable to the genetic engineering biotech techniques that were emerging at the time. So it was an interesting challenge to try to bring this genetics to sort of make this industry even better. Industry that was just starting up.
Interesting. What initially inspired you to get into this area of research?
Yeah, the inspiration was definitely, I wanted to save the world. So like a lot of people in my generation and probably yours, huge environmental problems facing us. And the thinking was that we need science to tell us what to do, how to do it better, how to do it more efficiently. And so that was my motivation, is to try to contribute to producing wood, producing trees in a more environmentally sound manner.
Sweet. If you don't mind me asking one more question, how long have you been doing this type of research?
This is my 40th year at Oregon
really?
State.
Wow.
Yeah, and I got my PhD in 1985. So I've been at this a while.
And did you start at Oregon State, or?
I've gotten degrees at other institutions, starting on the East Coast and then coming out to California, and then came up to Oregon State after I got my PhD for my first faculty position.
Okay, really? Well, sweet.
Are there historical examples of how humans have modified plants before modern biotechnology?
Yeah, one of the things, I have a class that I've taught for many, many years here about biotech and society and the interactions thereof, the scientific potential, the social issues regarding acceptance and how it can have value. And one of the things we start out with is a history of genetic modification. And so we've been doing it for 10,000, 15,000 years, depending on how you want to. It's starting with wheat. Is it starting with domesticating dogs? So where exactly does it start? Is it a few thousand years ago when corn started to becoming? So corn doesn't exist in nature in an edible form. Comes from a wild plant in Central America. And so that, in a sense, is where genetic engineering might have started, because corn is so radically different than its wild progenitor. So it's been going on, particularly in agriculture, for thousands of years. We could also talk about, beer and wine and the microorganisms that ferment it that have also been modified over the years. So I always like to have students understand what we've done and how radical it is, if you want to call it that, using conventional techniques. And so genetic engineering, gene editing is new ways of doing things that we've done before. In some ways they're distinctly different, and in some ways they're distinctly just the same, with just a different method.
Now, Steve, keeping in mind our listeners will vary from someone like me, with about an 8th grade level of knowledge of genetics and genetic modification, to perhaps people with deep knowledge of the subject. How do you describe to people the mechanics of genetic modification?
So, prior to the modern era, where we're doing what we're calling genetic engineering, we modified DNA radically, but we didn't pull it out of the cell and study it and sequence it and maybe rearrange it and put it back in. We modified it in the cell by selecting unusual mutant plants that occurred in nature, or we irradiated them, or we made hybrids between species on different continents that wouldn't mate naturally to try to produce lots of diversity and changes in the DNA. But it's happening inside the plant. In this era, we have the scientific technology to pull the DNA out and these days rather efficiently sequence it. We can determine your genome for a few $100s in basically a few hours. And that was, of course, unthinkable 50 years ago. So now we can study DNA with a rate that is unprecedented, and we can also see how it might be better for a particular purpose, how the natural DNA of wild or domesticated plants could be modified, but it might take a long time through conventional breeding. Or if we want to move one gene from one species to another for some reason to enhance nutrition, give it other properties, that we could do a horizontal gene transfer. That would be essentially impossible with conventional breeding. So I haven't really told you about how the mechanics of how it works, have I? So I'll just say that once you have DNA you want to put into a plant, you have to somehow get it in a cell, and then that cell that's modified in some way has to turn back into a plant, into an organism. And those are two problems. They're a set of separate problems. So we use this thing called agrobacterium. We can talk about more as you like, to put it in because it's a natural plant genetic engineer. That's how it lives. It infects plants with its DNA. The plants make a gall, which makes a nice home for the bacterium and feeds the bacterium. That gets it in, and then we usually use things like plant hormones to get those modified cells to regenerate, to divide and start to produce a multicellular organism. And so there's a lot of plant physiology that goes into how you do that latter step. And there's also a lot of different kinds of strains of agrobacterium and ways to use it and modify them and modify the genetic engineer itself. In fact, that's a lot of what we do in my laboratory these days, is find better strains of agrobacterium, figure out how they work, and modify them for higher efficiency.
Interesting. How does agrobacterium modify plant DNA, and how does this compare to other forms of genetic engineering or modification? Can this modification be considered natural, in your opinion?
So like I was starting to say, agrobacterium is a natural genetic engineer. There's no question about that. It's in nature, and it does its living by putting its DNA into a plant and getting it to turn into a home for the bacterium. So it is natural. And in fact, now we sequence genomes of everything, right? Like I was saying, it's cheap and easy. So we have the genomes and thousands of plants. And when you look at it on a computer, you see agrobacterium has actually inserted DNA into plants millions of years ago that became part of their genome that just evolved with the plants. So it's really been around. It's really been doing this thing for a very, very long time. And so what we're doing is taking this natural process, and it does it very efficiently because it's evolved to do it efficiently.
Yeah.
So we take that and we tweak the gene so it doesn't make galls anymore. We don't want a home for the bacteria. We just want to use it to transfer genes in so we can get genes in for whatever trade or scientific purposes and have a healthy, normal plant.
Sure.
The other ways of doing this are more physical, like you literally can bombard DNA in. There's a thing called the gene gun. So whenever I tell people about this, you aren't You aren't serious, are you? You're shooting? No, we're serious. People sometimes shoot DNA into plants, into cells. You have sort of micro shrapnel that's coated with DNA, and you literally shoot it in at high speed, penetrates a few cell layers deep, and the DNA dissolves off, and some low rate it goes into plant genomes and becomes a part.
Interesting.
So people don't use that much because it's so brute force. So agrobacterium is much more efficient, and when genes come in, they tend to be much more intact, not pieces of DNA shrapnel. So that's the preferred way to do it all over the world, every crop.
And I would assume using agrobacterium, it's far more predictable than
Exactly.
something like the gene gun.
It is, yeah. You grow it and culture it in a certain way. You induce the gene transfer. We know what chemicals, so an agrobacterium in the environment senses wounded plants. It can only transfer DNA when there's a little micro wound. So it senses that by the chemicals plants exude when they're wounded. So genetic engineers put that chemical right in the medium. We don't wait for the wound. And so that turns the bacterium on, literally, starts transferring its DNA. So it's much more efficient.
One just follow-up question I have as far as gene editing goes. When did humans discover that we could deliberately manipulate this process in natural ways? But when was this something we were truly focused on, or has it always been in some way or another?
You know, as people started to understand agrobacterium better, in terms of how its genetic mechanisms work, we're talking about the 60s and 70s.
Okay.
When we really understood it enough to sort of manipulate it and use it to our benefit. And then the first genetically engineered plant ever in the world was 1984, 85. And you may remember, I started in 85.
Yeah.
So here I am, this young professor trying to figure out what I'm going to do. And I say, wow, here's this potential. In fact, everybody in the forest industry and agriculture was saying, wow, how is this going to have an impact? How can we use it? How can we advance it? So I was just happened to be part of that trend. Then on top of that, people started growing these poplar trees so the first tree that was ever engineered was 1987, about 2 years after the first plant. And so here we had evidence that this is going to work in poplar trees, like we're starting to grow in Oregon on a big scale for pulp and for energy. And so I said, wow, can we help accelerate that process, make the growing of poplars and their breeding even better? So that's about the time frame, mid 1980s for that.
Okay.
Yeah.
And I'll leave the next question to you, but just one more thing. During your time in research, how has your perception of this research changed or evolved over time? Or instead, how has public perception of this evolved? As you, in this position, have definitely grown to learn more about it, and these processes have gotten a lot better.
You know, it's been a huge swing of the pendulum that I've seen during my 40 years.
Sure.
It seemed like in 1985, there was so much excitement that this was sort of the answer to every problem.
Okay.
And it in no way is the answer to every problem.
Yeah.
But that's the way it seemed. We were giddy with possibility. And then we sort of turned where there were some accidents in sort of how some things were rolled out. And some of the first products in agriculture were not really feel-good products. They were things like herbicide-resistant plants to help farmers control weeds, which didn't appeal to the average consumer. And the notion of an herbicide on my plants, my food plants, doesn't appeal in general. So for that and other reasons, there was a lot of pushback. And then a lot of the big environmental groups decided that they didn't like it very much and had major programs sort of against biotech. So then it became the thing that's responsible for all our woes as compared to the solution to all our problems. There was really pushback. And so we saw the erecting of very, very strict regulations, controlling research and application. We saw, as Scott and I were just talking about, we saw rules in forestry and in agriculture against use. So any organically certified food cannot use any genetic engineering, period. Any a certified forest product cannot use any genetic engineering. So it sort of turned where people were protecting themselves from it rather than adopting it. So big change. So in my career, I didn't foresee the pushback, and I also didn't foresee the value, the economic value of having a weird GMO-free label.
Okay.
So things have really changed. Yep.
Would you like to go ahead and ask the next one?
Yeah, thanks, Tegan. And this may tag on to what he was just asking you, but what are the common misconceptions about natural versus modified crops, and which of these misconceptions you encounter most often?
Yeah, and as we talked about earlier, the misconception that all our food is natural. And we talked about how they've been intensively bred for thousands of years. They've been moved all over the world. Potatoes and corn and soybean. The main foods we eat don't come locally. So people like to eat local. Well, that if that involves the sources of the actual crops themselves, you just can't do it. So that's one misconception. And then my sister and educated people, but not out of science, have no idea of the modification of our food system. They just don't know.
Yeah.
And so that's important they understand almost everything we eat is intensively modified. So I think people, and we know from many opinion surveys that people are afraid of things labeled as chemicals and unnatural, and they prefer what's considered to be natural. So we know that things in nature can be extremely hazardous and dangerous, whether they be natural diseases like measles or whether they be natural toxins in the environment. But people, we have such a high standard of living that people now perceive the main threats are from science itself or, corporate agriculture and the food system. So people like what's natural. So anything that smells unnatural is suspect. And in fact, our regulations really start from if it's natural, it's presumed to be fine and safe. So all of conventional breeding, for example, is assumed to be fine and safe, no matter who you hybridize or clone or mutagenize, whereas all genetic engineering is assumed to be hazardous until you prove elsewise. So it's deeply embedded in both our psyches and our legal systems.
Okay, interesting. I think you've just touched on this, but maybe from more of a context of climate change. Do you think these misconceptions affect the ability of society to use genetic engineering technology to help plants adapt to rapid climate change?
There's no question about it. It's really a struggle to think about how you could use these techniques of genetic modification in a timely manner, or to be able to use them not just in the major food crops where you have, giant multinational companies investing billions in development, but where you might want to develop, for example, a emerald ash borer resistant ash for Oregon, which is a public interest thing. There is not the money to overcome the regulatory hurdles in a timely manner. There's not the money to invest in the research in a timely manner. It's almost irrelevant. So my feeling is that our regulatory gridlock, plus the market obstacles we talked about earlier that make it, are so great. They've taken this very powerful science and technology and made it almost irrelevant. So it's chilling, but that's where I think we're at.
Interesting. Do you think it's possible to get most people to support genetic engineering rather than fear it is an unnatural or unethical process? And how might this be done in your perspective?
It's sort of a chicken and an egg thing.
Sure.
Where I think the main way, the surveys I've seen of, there's a part of the American public which, doesn't want GMO, doesn't want chemicals, doesn't, wants, maybe that's 20% or so, depending on the opinion survey. But the large majority of Americans are pretty pragmatic. And if you can produce a heat-tolerant tree that might survive, a heat wave, whereas non-modified trees would, and you could show it to them, I think they're going to say, cool, let's do that. So I think if we can get products out that show the benefits, I think people will accept it. But one of the big problems is just the pipeline is has so much friction in it for the reasons I mentioned, market and regulatory, that, like, for example, forestry companies around the world, I only know of one that's making a serious investment in this kind of biotechnology.
Sure.
And that's because the companies they don't see that these investments will pay off in a time frame that's relevant.
Okay.
So they would like to, no question about it, but it's a difficult proposition. So so how you would change people's minds, in short, is to produce products that show them. And we have some of those out there already.
Yeah.
We can talk about some of that, but we'd like much more of that, particularly things that appeal not just to corporations and farmers, but to the more general public.
Okay.
Yeah.
And then as far as that goes, here at Oregon State, with your research, do you experience a lot of hurdle as far as funding goes, just trying to continue with these projects that you're interested in, you're really trying to move forward? Are there often, barriers due to financial reasons?
Well, to get funding to do research,
Yeah.
Tegan, requires you write proposals generally to federal agencies
Sure.
to support it. And so you may know now we're in a period where everybody's wondering.
Yeah.
I wrote a fantastic proposal, I assure you, to USDA a year and a half ago that I think is really beautiful.
Yeah.
And it's just sitting there. And we have no idea if it'll ever be funded. And then we have grants in place that we have no idea if they're going to be pulled back.
Okay.
So the whole funding situation is very uncertain and very, very scary right now. So on the other hand, the Department of Energy has recently reasserted that we need much more efficient genetic engineering methods if we're going to have biotech modified energy crops at all.
Okay.
So they're really saying, Steve, the kind of thing you do, we want to pour money into. So that's also happening.
Yeah.
I just wrote another fantastic proposal to Department of Energy a few months ago to do this very ambitious genomic / genetic engineering work. And so it's sitting in Washington, and I have no idea if anything's ever going to happen with it.
Interesting.
So it's a crazy scene right now, in the United States of America, where we don't know what the federal government's going to do for science, whether they're just going to break it down by cutting or whether they're going to make targeted investments. So I'm scared, and all the people who work for me are scared about their livelihood.
Yeah.
Yeah.
I bet it's a scary time as far as science goes.
Yeah.
Now, a little final wrap up with this question is, now when you write a proposal like that and it's sitting there, can you use these proposals in, a few years from now if it seems like a time that those can go back out, or would you have to completely rewrite them, completely redo the idea?
Science moves fast.
Yeah.
And so if I sent in a proposal I wrote 2 or 3 years ago, it would be laughed out of the room because you wouldn't be using the most up-to-date techniques.
Sure.
You wouldn't be citing other key literature that's come about. So no, you really have to hopefully not start from scratch.
Yeah.
But you'd really need to put in probably, at least 30% of the effort again. And writing these big proposals, when you put them all together, they're 100 pages. They're 15 pages, single-spaced, of hard science.
Yeah.
And it takes a lot of effort.
To rewrite.
So yeah, no, we can't just keep them on the shelf and send them in.
Oh, shoot.
Yeah, Steve, you've already addressed this in some ways. This more about how plant transformation techniques might help address climate change related impacts. For agriculture and forestry, you gave the one example of a tree species that could be resistant to, say, our next heat dome. Are there other examples you can think of how these techniques might help, again, address climate change related impacts?
Yeah, one thing climate change does is it tends to change and promote pests and pathogens. And we know a lot about the genes that are involved in resistance to different pests and pathogens. So that's not directly temperature, but that's something that deals with a consequence of climate change, spreading pest populations.
Say drought and stress, huh?
Drought and stress, a drought and heat stress are more complicated traits. Often pest resistance has a simpler genetic basis. BioTech is a powerful method, but it's not a replacement for conventional breeding. Where you have so many different, you have about 30,000 genes in a genome, and genetic engineering tends to work with a half dozen.
Oh, wow.
So you need the kind of conventional breeding foundation. So it's some combination that will be most useful, putting those together. And as I mentioned, the problem is that one of them is highly controlled by the government and these market forces, and the other one is really freedom to operate. When you make a new hybrid,
Conventional techniques.
you make a new hybrid, you just plant it in the world, and you don't got to file any reports or get permission or get sued by Greenpeace or anything like that.
Last question that I have here is, and I think maybe this would be a recap for you because it certainly seems redundant, but where do you see the greatest opportunities and, of course, the greatest challenges for your field of research in the next, say, 5 to 10 years? We've got the funding, we've got the regulations. Is there something else you haven't touched on?
You know, one of the challenges is, I mentioned earlier, doing genetic engineering requires putting genes into cells and getting those cells to regenerate. And it turns out in trees, actually many crops, but trees are probably the worst, that's really hard. So what you're essentially doing is taking like a cell from a leaf and convincing it should start making roots and shoots and become a new tree again. And it thought it was going to be a leaf for the rest of its life till it, went to the leaf litter and died. So you're trying to reprogram it. And that's really hard to do. And the fact that we can do it at all is a small miracle. That's something that's incredibly hard to do in animals, but it's also very hard in plants. And the biology of what happens during regeneration and how to kind of manipulate it so it works efficiently is something we work on in a lot of labs, trying to solve that problem. So that's a big challenge, to understand it and then to tweak it. So right now, if you told me, Steve, I've got $1 million, go make an emerald ash borer resistant ash, I'd probably have to spend more than $1 million figuring out how to do the gene transfer and the regeneration, because we don't really know how to do it and we don't understand enough of the science of it. So I'd probably even argue, Scott, I'll work on a different species that's faster, and then we'll come back to ash in 5 years. So I think there's a lot of science related to regeneration in particular, also agrobacterium and gene transfer. That's a big challenge, but I think we have techniques that are effective and we can make progress if we apply ourselves to it. The biggest challenge, though, is on the social side, on people. People who are afraid because it's unnatural and want to erect very strict barriers. There's a cartoon that was going around some years ago of a bike locked up in a bike stand with 30 locks on it. And the caption said, "Do you feel safer now?" And I kind of feel like that's where we're at. Yeah. It's just because of people's fears, because of politics, we've made it really hard to move forward. So how you change that, how do you change these laws, is really, really difficult. So, yep.
Anything else that you wanted to ask, Tegan?
My final question usually is just, if you were to leave, the viewers, the listeners, with anything from what we discussed today, or given your research, and you want the world to have something to think about, what would that be? I know it's kind of a broad question, but there's a reason you're doing this. And what would you like others to just remember?
I guess the first thing is, I'm a scientist. I've signed on to this because I believe it's the best way of knowing the world, free of your biases and ideologies.
Sure.
So I think we need to invest in science and keep doing it. And it's also not something you can just turn on and off. If you stop investing in science for the next 5 years, you're not going to have trained people who can really do it.
Yeah.
A lot of people already are leaving the field because of what's happening at federal agencies and with grants. So you got to keep investing in science and investing in the workforce. The other thing is we have to figure out how to get out of our own way better, which I've talked about several times. So that's a real challenge. In a paper I wrote last year, we need sort of biological innovation and we need social innovation. Yeah. We need to do things smarter and faster and more science-based. And we haven't quite come together to figure that out. And so I hope we can do that. Otherwise all the great science won't get a chance to do much good.
And with that, another very brief question is, I think public distrust in the scientific community is, really high right now.
Seems it's been growing lately.
How would you suggest moving forward as far as like science communication goes? Does it take someone like me who's not super familiar with the science but is communicating and helping people like you communicate these things to the world? Or do you think that scientists have the power to communicate these things themselves and really, spread that information?
I think in the world we're in, I think scientists need to be dedicated to communicating with the public.
Sure.
And that's the National Science Foundation. Whenever they give you a grant, they have a thing called Broader Impacts, where you need to bring students into your lab. You need to go talk at the garden club. So that's already kind of wired into the system that scientists need to get out there. But there's so much out there now. You need to be smart about it, kind of like you guys are doing and we're trying to do in our NSF-funded project. You need to use the tools. the videos, the social media. You have to be fun and creative and artistic about it because there's just so much other stuff, and including a lot of bad, scientifically bad stuff.
Just cut through the noise with.
I think scientists have to be there, but they really need a team.
Yeah.
And we need to invest in it so it's out there. I think people still, when you look at ratings of credibility, scientists are still high. It's come down over the years, but
But it's not at the
compared to others.
Yeah.
Compared to others, people need science. They do, and they know that they do, and we have to keep earning that. And of course, the things that we're doing by like going against science, like going against vaccination, people are getting measles in record numbers. So people will see consequences.
No good's coming from that. And eventually people will realize that.
It may take some pain.
Yeah.
Maybe a lot of pain.
Well, sure.
Yeah.
Well, thank you for that perspective. And I think, as our podcast is funded by the NSF, just broader impacts, and that's the whole intention, is to try to bring this knowledge to people who might otherwise not know.
Yeah.
But yeah, thank you for all of that. And I'll leave you with the final little bit.
All right, yeah. So for the In the Woods podcast, we always wrap things up with a few questions in what we call a lightning round. And the first question is always, what is your favorite tree species?
Well, it's got to be poplars. It's
gotta be poplar.
So that's like cottonwoods and aspens. They're so beautiful, and they're such great scientific subjects. So yeah,
Okay.
easy.
Easy. I thought it might be. And then the second question, what's a unique tool that you use that helps you with your work?
The answer there has to be CRISPR.
Okay.
This is this thing that allows you to change any specific gene in the genome with pretty much 100% efficiency. Wow. It's just extraordinary. And it works everywhere. It works in every organism. It's very, very broad. So thank you, CRISPR.
Okay. And if people want to look that up, that's CRISPR.
Right.
Right? Missing
Yes.
the, okay.
People have them working on CRISPR apples and CRISPR tomatoes. It's true. Fun as it is, but the technique has been applied.
That's amazing. Okay. Lastly, what other resources can you share for a listener that would like to learn more about your research?
The best thing would be we have a good website at Oregon State. You could just Google Steve Strauss, Oregon State University. We have all the presentations my students, my postdocs, myself have given, and you can just see what we're doing and what we're saying, including some of the radical things about changing some of the laws to allow innovation to go forward. It's all written down, all in a bunch of very accessible PowerPoints. And so and you'd also learn about our values and our principles we operate by. It's all on our nice Oregon State website, so have a look.
Excellent. Yes. Yeah. Steve, thanks so much for spending some time with us today and for sharing your knowledge with our listeners.
Scott, thank you very much. Tegan, thank you. Really enjoyed the conversation. And unfortunately, Tegan, our generation is leaving your generation with some huge challenges. All I can say is I'm sorry and good luck.
Yeah. Well, thank you. And just one final wrap-up question. I think there are a lot of younger people, younger scientists who want to get into this field and are probably very interested but unsure. What advice do you have to them? I mean, I'm willing to bet a lot of people who see this are very motivated hearing what you guys have to talk about, but aren't sure where to start.
I'd just say if you see something that excites you in the news or in a Google search, don't hesitate to contact the scientists at the lab and say, hey, how do I get involved? We've trained hundreds of students who come in our laboratory and can make some money and learn, and it's set up a career for many of them. So don't hesitate to get involved.
Well, sweet.
Yeah.
Well, thank you. This has been a great conversation. This has been BioTech Talks with In the Woods. It's been great having both of you on today, but yeah.
Thank you, guys, and go Beavs.
Go Beavs. Yeah. Sweet.